So I guess to talk about that pancreatic cancer advance, we may need to go back a little bit into and briefly explain how mRNA vaccines work, because I don't think we've.
No, we haven't covered the vaccines yet. But if we're there, I mean, yeah, because the vaccines, I think the public scare and the disinformation of the anti-science movement came about RNA mostly came around the time of the COVID vaccine, and it only gotten worse since. But so yeah, I think the COVID thing, or not necessarily the COVID thing, but just the vaccine thing and the mRNA vaccine. I don't want to say technology, but maybe like technology based on the RNA, I think would be a very good starting point, maybe to get where we're at now and how we're going to move forward in science to better understand the applications of RNA.
Yeah. So if we take a step back from what I was talking about, like splicing, and we go back to just the idea that mRNA is a message that contains the information needed to make a protein. So for more traditional vaccines, the ones that were used prior to the mRNA vaccines, those were based on either using an inactive version of a virus or a protein that comes from the virus. And then that would be injected into our arms, and our immune system would recognize it as something that's like a stranger, and would produce a memory of that protein so that the next time it encounters it, let's say if you're infected with the virus, it recognizes it as being bad, and it destroys it. So the idea of the mRNA vaccines is the same, but instead of right away giving the worker the protein, we're instead injecting the message. And then our own cells will use that message to make the protein, and our immune system will mount a immune memory against that protein. One of the reasons why this is like a really valuable advance is that proteins and inactive vaccines or inactive viruses can be really hard to make in large quantities, whereas that it's actually pretty easy to make large amounts of RNA. And so if you inject the message, this can be read into protein by our cells and generate an immune memory, so that the next time you're infected with COVID, your cells will sort of recognize that it's something bad and fight against it. So there's sort of two things that branch out from that. The first thing is that there was a lot of work that was done prior to the pandemic to get to the point of being able to develop these mRNA vaccines. So when the media was saying that, it's amazing that the vaccines were developed in 9 months, this was only possible because of decades of research that happened before. So, for example, the two researchers, Katalin Karikó and Drew Weissman, who won the Nobel Prize in 2023 for their discovery that contributed to the development of the vaccines, that discovery that got them the Nobel Prize was actually made in 2005. And what they discovered is that if you just inject normal mRNA into our cells, our immune system will see it as a stranger and will destroy it before the message has any chance to be used to make the protein, which is what is really needed for immune memory. So what they figured out is that if you modify the chemistry of the RNA a little bit, so as if you were adding accents to the letters, for example, they're sort of unrecognizable by our immune system. And so this allows the mRNA message to be present in our cells long enough to make the protein that will mount the immune response. And then after that, the RNA is destroyed.
May I? Basically, is it prior to this discovery, is basically our system shut the messenger? We say often don't shoot the messenger, but basically our bodies saw a messenger and they just shut it down without ever getting the opportunity to read the message. But thanks to that discovery, basically our body says, oh, there's a message coming in. Let's get the message, read it, and then being able to understand it, because it's basically the instruction manual on how to fight said disease. Is that a good metaphor?
Yeah.
Okay.
Yeah. And so going back to pancreatic cancer, we can use a similar approach to treat certain cancers, where here, instead of being a message that helps our immune system recognize a virus, we're instead using a message that will help our immune system recognize only tumors and kill or destroy those tumors. So the way that it works is there are mRNAs that are specific to tumor cells that are not found in our normal cells. So if we can identify those, the way that it was done for pancreatic cancer, you can generate a vaccine, so an mRNA that you can inject, and then our immune system will learn to recognize that message as being bad. And when it sees it, it will destroy it. But because it's in our tumors, it will destroy the tumors. And this is likely to have a lot less secondary effects than, for example, chemotherapy, where normal cells can also be affected by the chemical that is used to kill the cancer cells.
So it would allow to have way more targeted approach for specific kind of cancer. We're talking about the pancreatic cancer now because that's the one that have great scientific advances. I'm pretty sure many labs are working on other kind of diseases or labs, but it basically shows for those of us, for those our listeners, it shows that RNA is actually a good thing. It's a good thing to understand. It's a good thing for us. It's a good thing for the scientific community to work a better understanding RNA so that then it can benefit society as a whole, which science does across the board, but we're more focused about RNA today.